I actually want to go back to something that we have already talked about just briefly to mention that there's more than one way by which genes are regulated in bacterial cells. So obviously operon systems are one, and this represents a mechanism of genome organization in bacterial cells. The other thing that we find, you may recall we talked about at promoters, we have these little bean-shaped guys or gals, and the RNA polymerase. What's this little bean-shaped thing called? And it recruits the RNA polymerase to these promoters. It's a Greek letter. It looks like this. Sigma factors. So, and one of the reason why I bring this up is that there are 7 or 8 of them in E. coli. The main takeaway point with them is that each of them is controlled or expressed under different conditions. So for example, as a cell transitions from log phase into stationary phase, we get increased expression of the sigma S gene as cells experience heat stress. So if we take E. coli and start to grow them at 46, 47 degrees Celsius rather than the 37 that they're typically used to, we start to find expression of sigma H. If there's envelope stress, so if ethanol is present, sigma E gets expressed. And each of these recognizes different minus 35 and minus 10 regions of those promoters. So each of them controls expression of a different set of genes. This is one way in which bacterial genes get controlled. The other is with using mechanisms like we find at the lac operon, where binding of additional proteins can help to either block the RNA polymerase from proceeding, or can actually help to recruit the RNA polymerase to the promoter. Okay? So I don't want to start with sitting here and talking about the operon. Rather, what I want to do, and it looks like we're kind of light today, but maybe that's in part in contrast to what we had on Wednesday. Oops, cancel that. I'm just going to leave this up, and what I want you guys to do is, there are 4 conditions again in which we typically look at the lac operon. And we look at the presence of 2 carbon sources. Conditions when glucose and lactose are present, when glucose and lactose are absent, and when one or the other is present and the other is absent. What I'd like you to do, go back to the homework assignment, the wiki. I've put some notes in some of them. And also, right now, I'm going to also post up the actual results that I have listed. And I want you to go through and start to look at trends again. Not everybody took the time to do that before today, but I do want to make sure that we have time. And that's the big focus, or how we'll spend the first 10, 15 minutes of class, is identifying trends here. So, oops, not that. Okay, make that available, and nope, cancel. Damn it. All right. Oh, for goodness sakes. So here are the answers as I see them in each of these 4 conditions. In the presence of both of these carbon sources, lac I is bound to the operator, cyclic AMP is not present, CAP would not be present either, and so we might get some binding of the RNA polymerase, but it's going to be really weak. So if you're thinking about this in just binomial yes or no answers, the answer here is no. If you're thinking in shades of gray, yeah, there's some, but there's really little transcription that occurs in this case. In the presence of glucose but the absence of lactose, we find lac-I being bound to the operator. Cyclic AMP and cap are not present, and we get no binding of the RNA polymerase and no transcription. In the absence of glucose, but presence of lactose, lac-I doesn't bind, cyclic AMP is present, cap is present, RNA polymerase binds, we get transcription. And then last, in the absence of both, no transcription is occurring, but lac-I is present at the promoter or at the operator, so is cyclic AMP and cap. So those are the basic conditions. Here's what I'd like you to do, 2 parts. 1, draw out one of these conditions. Draw out what would the operator and promoter actually look like under one of these conditions. All right? So is cyclic AMP bound to cap, and are those 2 bound to the DNA or not? Is RNA polymerase there? Is lac-I present or not in these conditions? And then I want you to, with somebody else, identify at least 2 trends that you see. And by trends here, I don't mean, oh, when lactose and glucose are present, lac-I is not bound. That's okay, but what I want you to do is actually go between these rows. Look for commonalities between rows. So as an example, you may not use this after we use this in class, or right now. When lactose is present, so first row and third row, what's true about lac-I? It doesn't bind. In other words, lac-I's ability to bind to the DNA, one way that we might say this, it's dependent on the presence of lactose. It only binds to the DNA when lactose is absent. Okay? That's kind of one of those cross-cutting trends that I want you to take a little bit of time identifying. So 2 parts. Draw one of these conditions, and then with somebody else identify a couple of trends. I'll give you about, well, let's go to 11:15. And then we'll come back as a group and spend some time talking about this. Okay, what the hell is this thing? What is the lac operon, and why should we even care? So let's start with the probably most basic of these questions, not meaning it's the easiest to answer, but it's a good starting point. What is lactose? It's a carbohydrate. Right? Phenol red tests. We did phenol red tests to test for the ability of microorganisms to ferment 3 different carbon sources. One was glucose, one was lactose, and then the third one was sucrose. Okay? So lactose is a carbohydrate. In the presence of glucose and lactose, or let me back up. Carbohydrates are used for what? Like glucose. How did we talk about glucose this semester? What does the cell use it for? Mumble, mumble, grumble. Energy in the form of ATP. Lactose is used in the same way. Purpose is to make energy for the cell. Now the cell's not going to waste the energy to make or to turn on genes that are used for metabolizing lactose and glucose at the same time. Bacteria like E. coli, the default is if glucose is around, that's what they'll break down first. But at some point, there's only a finite amount of glucose within the media that we provide cells. And that glucose is going to produce a lot of ATP. Well, when energy is burned, or energy is used, ATP releases a phosphate, forms ADP. If there's no glucose, we can't regenerate that ATP. So ADP will be broken down to AMP. And that ultimately will be circularized to form this cyclic AMP or CAMP. This is what some researchers call an alarmone. It's an alarm that acts kind of like a hormone to bacterial cells. It's a sign that energy levels are really low and that it has no way of producing ATP. Kelly.
What's the second thing that's being released after the ADP?
Sorry, that's supposed to be another inorganic phosphate.
Oh.
Yeah.
I don't know if it was like an R.
No, it's not supposed to be, but it definitely looks like it.
Okay.
PI. There we go. So this is an alarm. Energy levels are low. What does the cell do in return? If energy levels are low, it's got to find a different way to make energy. How does it do that? At least in this context here. Changes the carbon source. So if lactose is present, lac-I comes unbound from this promoter, from this lac operon, and that allows for gene expression to occur. And the genes that get expressed, the LAC-Z, the LAC-Y, and the LAC-A, basically their function between the three of them is to bring to bring the lactose into the cell, and then to take that disaccharide and break it down into two monosaccharides, galactose and glucose. And at that point, then they can replenish their ATP stores. Okay. So this is kind of a backup mechanism, right? Think of it in the same way as you would maybe like power to a house. If your power goes out, it's always good to have a backup generator so that you can continue to run the energy. That's what's happening here. So organisms that are capable of fermenting lactose, they have this system. They have the ability to perform this breakdown of lactose. So that's the big picture. Okay, that's the big picture idea. If we look at what's happening specifically, well, let's start with trends. What are some other trends that we see in here? And I'll just go ahead and I'll list them as we go through. Say that again, Emma? Okay, so cyclic AMP is only there if glucose is gone. And that fits with what we've just kind of modeled here. Glucose is gone, the cell's going to burn up all the energy it has. This gets produced, cyclic AMP gets produced at that point when glucose is absent. Absolutely. Perfect. What other trends are there?
When the cyclic AMP is present, it binds to the CAP, and then that binds to the promoter.
Perfect. So cyclic AMP so what Kelly just said was cyclic AMP will bind to CAP. And when that happens, CAP binds to the promoter. Can CAP bind to the promoter without cyclic AMP? No. This is similar to something we've discussed. You may remember at one point this semester when we were talking about metabolism, we were talking about enzymes and ones where here's our enzyme, here's the active site. And then if a molecule binds to it somewhere else, it changes that active site so that it no longer can bind to its substrate. What kind of molecule is that known as?
Non-competitive inhibitor.
Yeah. This is functioning a lot like a non-competitive inhibitor. What we could think of CAP or cyclic AMP as, we can think of this as a non-competitive activator, actually. It's activating a function of CAP, or it's changing the shape of CAP in a way so that it now can bind to the DNA and function like a transcription factor, a protein that's going to enhance the transcription of these genes.
So instead of it changing the shape to not fit, it changes the shape to fit?
Exactly. Exactly. So it changes the shape of cap so that, I'll draw this a little bit differently. Erase this real quick. So I'll just make this red dot our cyclic AMP. Here's our cap. So it takes this would be the active state, where it can bind to the DNA really well. If it's not bound, basically forces the cap to change shape so that it can't bind to the DNA. Exactly. Good. What are other trends that exist? When do we get transcription? Alana? That's when we get the strongest, right? Absolutely. Yeah, you might get really weak. So the one thing that's common between what you just said is that in each of those cases, lactose has to be there in order for this to be turned on. In other words, the cell's not going to waste its energy on producing these mRNAs, and producing mRNA takes a lot of energy, and it's not going to waste the energy on translating them unless that carbon source is present. Okay? What else is true about that? Let's kind of go with this. Transcription only occurs when? RNA polymerase has to be present. What else? Take a look at the rest of those columns. We know lactose has to be present. And if lactose is present, what happens to lac-I? Can't bind, inhibited. Lac-I can't actually bind to the DNA at this point. So let's go off of what we just said about cyclic AMP. It's a non-competitive activator of cap. Another way we could describe lactose. It's a non-competitive repressor or a non-competitive inhibitor of lac-I binding to the operator. So, let's draw that out real quick. So if this is our, again, DNA promoter region, lac-I, the default state. Bind to the DNA. If lactose is present, it changes the shape of lac-I in a way so that it no longer can bind. So this would be gone, which would allow for transcription to occur. In essence, what we have here are two binary switches. One that is dependent on lactose and one that is dependent on glucose. Okay, so let's take a look at those two switches real quick. Whoops. All right. So a system based on glucose and one based on lactose. What is lactose control here? Of all the proteins that we've talked about, cap, lac-I, RNA polymerase, what is lactose control? Or which one does it affect? It affects lac-I. So lactose will control the lac-I protein. Glucose controls what? Indirectly. Glucose controls cyclic AMP levels, which regulates what? In other words, what does cyclic AMP bind? Good. binds cap, and then allows for it to bind to the DNA. Okay. These two bind to different regions on the DNA. This arrow here again is just representing where transcription would start. The LAC-Z, Y, and A genes would be to the right or downstream of this. And then upstream in this promoter and operator region are where the cap would bind, where the LAC-I would bind, and where the RNA polymerase would bind as well. We can think of these as two individual switches that are going to allow or prevent transcription from occurring. So if lactose is absent, LAC-I binds. Does this turn on, or does this prevent transcription from occurring? And take a look at if you need to, take a look at the chart again. If lactose is absent and LAC-I is bound to the DNA, do we get transcription? No. So this is what we would call an off state. Prevents that transcription from occurring. If we add lactose, that releases LAC-I from the DNA. This would be a state in which we now might allow for transcription to occur, or an on state. Cap's a little bit different. What is the cap will bind to the DNA when? When there's no glucose. Good. When cyclic AMP levels are high. Here, the binding represents an on state because cap is an activator of transcription. And without cyclic AMP, this represents our off state. So the only time that we get maximal transcription is when both of these are in an on state. When is that? What condition is that? In other words, when is the only time that we find cap bound to the DNA and LAC-I unbound from the DNA? Wow. Try that again. That would represent a state in which both of them are off. When there is no glucose, cyclic AMP is produced. That allows cap to bind. And when lactose is what? When there is lactose. Yeah, exactly. So when lactose is there, that removes LAC-I. When there's no glucose, that allows cap to bind. And the function of cap in this whole process is to recruit or to bring to this promoter the RNA polymerase. I liken the cap to a molecular beacon. It's basically serving there as a signal to say, hey, RNA polymerase, begin transcription here. In some ways, you can liken it to a sigma factor in this case. It helps to bring the RNA polymerase to this site of transcription. LAC-I functions a lot like a roadblock. It sits just in front of where the RNA polymerase binds. If LAC-I is there, the RNA polymerase can't proceed with transcription because it can't physically move past the LAC inhibitor. The only time we get transcription to occur is when this is removed. Okay, when we remove that roadblock, now RNA polymerase can begin that actual process of transcription. Let's try a review question, because I think if I say other questions, I'm not going to get any. I think it would be better to practice this right now. Does everybody have enough of what they need here, or okay. I just wouldn't want to erase it beforehand. All right. I don't want to do that one right now. Let's do this one. Which of the following is a trend of the LAC operon? So A, cyclic AMP is present when glucose is present. B, RNA polymerase transcribes LAC-Z, LAC-Y, and LAC-A when lactose is present. C, LAC-I binds to the operator when lactose is present. D, CAP binds the promoter when glucose is present. Or E, cyclic AMP is present when lactose is absent. All of these are wrong with the exception of one. So what I'm going to do is I'm going to give you a couple of minutes to go through it and identify the one that is wrong, or sorry, the one that is right. And then once you've done that, if you think you've got it, try fixing the others. Okay? Come up with a way to fix each of the other ones. And we know that those two are, one way we could say it is they're inversely correlated with each other. When one is high, the other is low, and vice versa. So A can't be the right answer. What else could we rule out? The last one? Is that what was that? Eliana? Wonderful. Why?
Because lactose has to be present for CAP to bind.
Well, so let's go back to our model real quick. We talked about this being two binomial switches.
Mm-hmm.
Have we talked about lactose being required for cyclic AMP levels? No. That's why I would say F or E there probably isn't the right answer. Because what is cyclic AMP based on? Glucose levels, not lactose. So the last one there wouldn't work because we haven't talked about cyclic AMP being directly dependent on lactose. Okay? It's based on glucose. What other ones can we rule out? We can rule out C. LAC-I binds to the operator when lactose is present. How would we fix that one to make it correct, Allie? Yeah. LAC-I only binds to the operator when lactose is absent. Good. So that leaves us with two, D and B. Which of those two is correct?
B.
Okay, good. Why is D not correct? CAP binds to the promoter when glucose is present. It's when it's absent, exactly. It's when cyclic AMP is around. B is the best answer here because lactose, when it's present, what happens to LAC-I? Doesn't bind, which allows for the RNA polymerase to begin transcription. This question here, at least the way that B is phrased, it doesn't tell you anything about cyclic AMP levels. Okay, so you have to consider this independently of that and just focus on the lactose levels. Okay. Shall we do one more? All right. This one is a bit more, this one's a little bit different. So the question reads, what might you expect to happen to the regulation of this operon? So how might it change if a point mutation occurred, a missense mutation, that on LAC-I that blocked its ability to bind to lactose? In other words, LAC-I no longer can bind to lactose. Why? Good. So if LAC-I is unable to bind to lactose, what's its default state then? How would it behave? Off. In other words, it's always bound to the DNA. That's great. Excellent. Which of these then is true? So is it A, LAC operon will function the same way, B, LAC-Z will never be expressed, C, the operon will be expressed in the absence of glucose, D, LAC-I will remain bound to the operon even when lactose is present, E, B, and D, F, C, and D. Yeah, E. So D definitely fits with what you said, Emma. We know that has to be in the answer. It's not A. By default, D negates A from being a correct answer. The operon would be expressed in the absence of glucose. Probably not, because we know that LAC-I is going to be there to inhibit transcription from always occurring. LAC-Z will never be expressed. That is true, because if LAC-I is bound, we will not get any transcription. Okay, so this one, E, is the best answer. What I'm going to do is I'm going to give you guys a quick minute to look through your notes on this section, because now we have kind of covered most of everything. I think this is the point where finishing off with a couple of questions might be beneficial. So I'll give you a minute to look through your notes, and then if we've got a couple of questions, we'll finish up.
But it's glucose too because the cancer's
Any questions? How about we finish with this? Unless there are any questions, let's finish with this for today. If you were to describe how the LAC operon functions, how would you describe how it works? All right, and by this I don't mean tell me what happens in all four conditions. That's not what I mean. Think about this in terms of trends. What are a couple of the major themes or takeaway points from the LAC operon and how it functions? So take a minute to think about that. Kylie, can you give me one major theme from this? Just the LAC operon. Describe it to me. How would you give me one piece of a description for the LAC operon? Phone a friends are welcomed.
Isn't it like the backup plan?
That's a great way to describe it. Backup plan. So if glucose, the primary carbon source for most microorganisms, if that's not around, turn on this. What else? Lexi.
It needs to have lactose to transcription.
Beautiful. No need to express it unless the carbon source that this is designed to metabolize is present. So it needs lactose in order to be expressed. What else? Let's try a couple of more. Allison, give me one.
Oh my gosh.
I know, I'm sorry. I'm an asshole.
Let's see. Say like glucose is preferred.
Yeah, glucose is preferred, that's great. That's a nice add-on to how Kylie put hers as well. One more. That wasn't a raised hand, was it, Taylor? I didn't think so. Okay. Paige, do you have one? So yeah, so maybe one of the ways we can describe that, tease it out a little bit, is just to say this is an example of how bacteria control gene expression or transcription. Okay, so we've seen two now. Choice of sigma factor. This represents a completely different system. Only turn on the genes when we need them to be turned on. Okay. So and that's kind of the broad way to think of this. It is a mechanism of controlling transcription. Okay. All right. The homework for Monday is just a quiz. You can take it as many times as you want to. We're focusing on horizontal gene transfer on Monday. All right. Have a great weekend. Enjoy.